Okay, so yeah, I'm very pleased to now introduce my dear colleague and friend, Dr. Cedric Neveu. He is coming from France, and initially he was trained as a plant parasitologist, but then he found that it's much more interesting to work with parasites from animals, and so he moved into the direction of parasitic nematodes of animals when he obtained a permanent position at the Institut National de Recherche en Agriculture, Alimentation et Environnement, or in French, in English, the French National Institute for Agroeconomical Research in the lovely Loire Valley. And he and his group are well known for performing very nice, elegant studies on the mode of action of anthelmintics, particularly, but not only, on nicotinic acetylcholine receptor agonists or antagonists in parasitic nematodes, but as well on the molecular mechanism of resistance. And yes, I'm very pleased that with this now also his group is somehow becoming a associate with Combar, and we very much look forward, Claude, now to your talk entitled Monitoring Anthelmintic Resistance from Phenotypic Assays to Molecular Markers.
Many thanks, Georg, for the introduction, and many thanks to you and Ioannis for the kind invitation to talk to you today about the molecular markers to phenotypic assays. So this work's performed in our team that is mostly dedicated to the search of mode of action and also search for new targets for the development of new drug. But I will give you some example about how our research could be also useful to identify some molecular markers. So I would go very quickly concerning the context, because as Combar participants, you are all aware about the importance of anthelmintic resistance, and Laura made a very nice introduction concerning the rather low efficacy now of PZ, and also for macrocyclic lactons, and in lesser extent to cholinergic anthelmintics such as levamisole, parentel, and morantel. So the first things I would like to address is, why do we need these molecular markers? First, they're going to be very useful in order to optimize the anthelmintic treatments. They can be used also as predictive tools for predictive potential cross resistance. And also they can be very useful to monitor spreading of allele resistance into the population. So this is not an exhaustive list, but at least these points are very important. I will try to give you some example about how we are identifying these molecular markers for anthelmintic resistance, using either candidate gene strategy based on C. elegans data, or using without a priori approaches, such as comparison of worms that are susceptible or resistant to anthelmintics. And these comparisons are performed at the genomic or transcriptomic level. And finally, we will see how to validate them both in the lab and in the field. So surely have only 15 minutes or something like that. So I will really focus my talk today on one class of anthelmintics that are the cholinergic anthelmintics, the one that remains potentially efficient to control both benzimidazole and ivermectin's resistant worms. And I will focus on levamisole, parentel, and morantel. So first of all, how do these drugs are acting on the worm? So these anthelmintic compounds, such as levamisole, parentel, morantel, but also a lot of them, such as oxantol, nicotine, dercontel, and so on, are activating or inhibiting the acetylcholine receptors from the worms. These receptors are pentameric ligand-gated ion channels that are involved in fast synaptic neurotransmission in both vertebrates and invertebrates. And their permanent activation or inhibition by the drugs will lead to the worm paralysis. And then the host perfectly knows how to get rid of the paralyzed worms. So you can have two different kinds of receptors. The first one are homopentamers. They are made of the same subunits. And the other one can include combination of different subunits, and they are the heteropentameric receptors. What is important to know here is that different subunit combination represent distinct subtype of AChR, and that the different anthelmintics can be selective for these different subtypes. So here, our working hypothesis was very simple. Because the best molecular markers are directly involved in the phenotypes, we decided to investigate polymorphisms that could impact the functionality of these receptors that are targeted by the anthelmintic drug. So I'm sure that most of you perfectly know that games that consist on identifying a combination of 5 different colors. And clearly, we are facing a problem with nematodes because there is a large diversity of AChR subunits in nematodes, much more than in vertebrates. So, for example, there are at least 29 different subunits in C. elegans in nematode model and 30 in Emanchus contortus. So, clearly, if we had to identify the anthelmintic targets just by testing random subunit combination, it would be like playing a super mastermind game with more than 30 different colors. So, clearly, just impossible. So fortunately, we have here 2 main models that could help us to identify the molecular targets. The first one is C. elegans. So in C. elegans, the good news is that this model nematode is susceptible to all cholinergic agonist anthelmintics. And clearly, screens for mutants that survive the anthelmintic exposure have been of major help in order to identify the AChR subunits that are involved in the corresponding receptors. However, clearly, there are some also major limitations with this model. First, C. elegans is not a target species, and clearly AChR subunit combination and pharmacological properties of his receptors can be different from the one from the parasites. So we also have the frog's eggs. This frog is an African toad, and this egg is a wonderful model for electrophysiology experiments. It is widely used to express nematodes AChR receptors made of AChR subunits. And by using the 2-electrode voltage clamp techniques, you can record some currents that are elicited by acetylcholine, but also the cholinergic agonist. Here you've got an example of a recording trace. So the receptors is expressed at the membrane, and when you apply acetylcholine, you observe this depolarization of the eggs. And the same thing with levamisole, showing that you have expressed the levamisole receptors into the eggs. So this model is extremely useful to decipher the subunit combination of the nematode AChR, but also to perform detailed pharmacological characterization of the receptors, and surely to investigate the role of the polymorphic AChR subunits that are potentially involved in anthelmintic resistance. So let's start with the candidate gene strategy. So in C. elegans, the selection of mutants that survive exposure to levamisole has led to the identification of 5 different genes encoding 5 different NAChR subunits. And these subunits are called UNC38, 63, LEV8, UNC29, LEV1. UNC is for the name associated with the loss of function of the gene. UNC is for uncoordinated, meaning that the worm that lacks these subunits doesn't move normally. LEV is for levamisole resistance. And what has been shown by Thomas Boulin in 2008, so an old paper, that if you express these 5 subunits together into the xenopus then you get a functional receptors that is sensitive to acetylcholine and that is responsive to levamisole. So demonstrating that these receptors potentially is the levamisole receptors in the model species. And also very importantly, it has been reported that UNC38, 63, and 29 null mutants are resistant to levamisole, parental, and morantel. So clearly suggesting that these receptors could be the main targets for the 3 different drugs into C. elegans. And these data laid a strong basis for the candidate gene strategy. And this is why we try to search for homologs of these genes into the parasites and try to identify putative polymorphism associated with resistance. So the first thing to address is that, was it possible to have a direct transposition of these C. elegans data to parasitic species? So orthologs of UNC38, 63, and 29 can be easily found in all nematode clades. And with the genomic research we have right now can confirm that these subunits are highly conserved all through the nematode's phylum. Whereas in sharp contrast, there are no homologue of LEV8, but also LEV1 in some case in many parasitic species that are sensitive to levamisole, parental, and morantel. So this clearly suggested that LAChR might have different subunit combinations between C. elegans and the parasites. But at least as a starting point, we started to compare what we had into our hands meaning 38, 63, and 29, in levamisole susceptible and levamisole resistant isolates from Emanchus, Deladosagia, and Trichostrongylus. So just by performing simple RT-PCR on cDNA from the 3 species, from levamisole resistant and levamisole susceptible isolates, what we've been able to show is that the complete coding sequence are expressed in both levamisole resistant and levamisole susceptible isolates. That when you look carefully at the sequence, you don't find any polymorphism or changes between levamisole resistant and levamisole susceptible isolates. But what we observed is that in our highly resistant isolates from Emanchus, Deladosagia, and Trichostrongylus, we identified an additional amplification product. So then we cloned it, and what we found is that this additional amplification product observed in the 3 species corresponds to a spliced variant of UNC63. And for Emanchus, we have raised some antibodies that are against the peptide that is common to both the full length and the truncated isoform, and we've been able to confirm in the levamisole resistant isolates that you have the co-expression of the full length mature subunits as well as a truncated isoform of the UNC63 gene. So at the end of the candidate gene strategy, what did we have into our hands? So first we had at least 3 potential LSHR subunits from 3 different Trichostrongylus species. And we have observed a polymorphism here that corresponds to the co-expression of both complete and truncated UNC63 protein in worms from highly resistant isolates. So this was a good starting point, but clearly it was very incomplete. So we needed for a complementary approach in order to identify other markers, and if possible, some markers that could be common to resistant isolates and highly resistant isolates. And also we need to identify the other potential LSHR subunits that are involved in the LSHR composition from the parasites. So what we did here is that we decided to perform a without a priori approach, where the main objective was to compare the transcriptome from levamisole resistant and levamisole susceptible Emanchus using cDNA FLP. That is a molecular biology approach that is very, very attractive because it will allow you to investigate expression polymorphism, but also transcript polymorphism, such as point mutation and insertion deletion events. So we performed these experiments using 2 resistant isolates and 2 susceptible isolates. And among the 17,000 transcript-derived fragments that were generated, only 26 presented a polymorphism between resistant and susceptible nematodes. And 15 of them were present in both susceptible isolates and absent from both resistant isolates. But even more attractive, 11 were present in both resistant and absent from the susceptible isolates. So potentially some very attractive resistance marker here. And among these attractive candidates, we've been very pleased and lucky to identify a fragment that is called HCO-RCR8B, because this corresponds to a spliced variant to HCO-RCR8 that is specifically expressed in levamisole resistance, and RCR8 is an NSRHR subunit that is closely related to LEV8. So suggesting that RCR8 would potentially replace the missing LEV8 subunit into Emanchus contortus, but also other nematodes levamisole receptors. This was also a very important result because we've been able to confirm that this spliced variant from RCR8 was also found in other isolates from our lab and specifically expressed in the levamisole resistant and absent in levamisole susceptible isolates. But this was also confirmed since by other labs on many other field isolates from Australia, North America, South America. So a nice and promising marker. And also very interestingly is that as observed for UNC63, the abbreviated isoform from RCR8 was co-expressed with its full length in levamisole resistant isolates. So clearly this was suggesting that the expression of some abbreviated isoform of the SHR subunits could constitute a response to the selective pressure exerted by the anthelmintic. And this is also further supported by the work performed on monopental, where Lucien Ruffner in Ronald Kaminski's group showed that Homunculus also responds by expressing truncated isoform of HCO-MPTL1, which is the target of the monopental in Homunculus. And this was also recently confirmed in field isolates by Andrew Coetzee on Australian isolates. So now the next step is, how can we investigate the putative involvement of this truncated isoform of splice variant into resistance? So here, let's go back to the Xenopus oocyte. What we did first is that we tried to co-express into the Xenopus oocyte the subunits that we had into our hands, 38, 63, RCR8, and N29. And we've been very lucky at getting a very nice and robustly expressed functional receptors here that is extremely responsive to levamisole. Here you can see that levamisole is acting as a full agonist on these receptors. So levamisole is more potent on these receptors than the C. elegans receptor. And clearly, it was for us also a major opportunity to test the role of the truncated N63. And by mimicking what we can observe into resistant worms, we co-expressed in the Xenopus oocyte all the subunits that are required to get the functional expression of the levamisole receptors, in addition to 63 at different ratio, 1:4:2 and 1:10. And what you can observe here is that the expression of the receptors is severely impaired when you had this truncated isoform. And this strong dominant negative effect on the receptors was something very important because it was representing at that time the first functional evidence for a potential molecular mechanism of levamisole resistance in parasitic nematodes. So then surely we tried to do the same thing with the truncated HCO-RCR8 by co-expressing truncated HCO-RCR8 with the levamisole receptors. But here, unfortunately, we didn't get any results. We didn't observe the dominant negative effect. Potentially, we hypothesized that's because there is a lack of transmembrane domain and that potentially also Xenopus oocyte might not be the best model to test the involvement of RCR8B in resistance. But clearly, what is important to have in mind is that the role of the truncated RCR8 in levamisole resistance remains to be established. So anyway, we were not able to play with the short one. So we tried to get some more information about the full length one, about RCR8. What is the role of RCR8 more precisely? So when we tried to identify the minimal subunit combination that give rise to the functional receptors, what we noticed is that when we got rid of RCR8 in the mixture, we completely lose sensitivity to levamisole. We make a new receptors. We make a new receptors that is able to assemble without RCR8. And this receptor is far less sensitive to levamisole. But in sharp contrast, this receptor was highly sensitive to parental. That was very, very attractive because clearly, because the difference between HCO-LCHR1, this one, and HCO-LCHR2 is the only presence or absence of HCO-LCHR8. So this clearly highlights the critical role of RCR8 into levamisole sensitivity. So indirectly, this highlighted the importance of the RCRB marker and also suggested that in sharp contrast with C. elegans, levamisole and parental could act on distinct receptors in Homunculus contortus. So why do we, how can we test the hypothesis of one target versus multiple targets within the worms? So what we did is that we tried to inject RCR8 into C. elegans LEV8 mutants, okay? And what we observed is that when you do that, it does not change the motility of the worms. The N2 are moving normally, the LEV8 mutants are moving normally, and the one expressing the RCR8 from Homunculus are also moving the same way. But when you apply levamisole, clearly the N2, the wild type strand, is paralyzed by levamisole. LEV8 is resistant to levamisole, and the transgenic line LEV8 expressing RCR8 will become sensitive to levamisole. So we restore levamisole sensitivity here. And exactly the same thing for parental, meaning that just by adding one subunit, we were restoring both levamisole and parental sensitivity, suggesting that in C. elegans, levamisole and parental could have the same targets, that is the LSEHR. But strikingly, when we investigate RCR8 in vivo into Emanchus contortus by silencing the subunits into L2 stage, what we observed was something very different. Because when you silence RCR8 into Emanchus, then the worms become resistant to levamisole, as you can see here. Whereas in sharp contrast, this does not affect at all the parental sensitivity. And this is something that is of major interest. Unfortunately, we are not able to silence also RCR8b into our levamisole resistant worms because it was not applicable, as the sequences are too closely related between the short one and the long one. But clearly, these results were supporting the hypothesis that levamisole and parental could have distinct molecular targets in Emanchus. And also of importance, when we were investigating some novel SHR muscle subtypes in order to get some new targets for the development of new anthelmintics, we identified a novel SHR muscle subtypes that we call the MSCHR. These receptors is made of 2 subunits that are only found in animal parasitic nematodes and that are absent in the free-living nematodes, such as C. elegans, but also absent in plant parasitic nematodes. And 2627 makes a functional heteromeric channel, so expressed in the muscle, and this receptor, surprisingly, is incredibly responsive to morantel, and in a lesser extent to parental. But what is very important here is that we got an additional subtype of receptors that is completely insensitive to levamisole. So when we expressed these receptors from Emanchus or from the distantly related parasites, Parascaris equorum, into C. elegans, we were able to show that expression of this MSCHR into C. elegans was leading to a drastically increased morantel sensitivity to the worms, and in a lesser extent to parental, so validating this novel muscle subtype as the morantel SCHR, MSCHR. So then with all this information into our hands, how can we speculate about the interest of truncated UNC63 or RCR8b as marker for predictive, as a predictive tools? Here I would say that it might not yet be the case. Okay, so we got the HCO-LSHR1 made of 38, 63, RCR8, and 29. We've been able also to show that these receptors was clearly obtained also in other species, such as Oesophagostomus dentatum. We've got, and these receptors is supposed to be the main targets of levamisole. We've got the second subtypes, HCO-LSHR2, that is also found in Oesophagostomus dentatum, the one that is lacking RCR8, and this one might be a preferential target for parental. And we've got this third subtypes made of 2627 that has been also confirmed in distantly related parasites such as Parascaris equorum. And this one might be a preferential target for morantel and parental. So now concerning the truncated UNC63, if we assume that the SCHR that includes UNC63 could be impacted by the truncated isoform, then we could predict a cross-resistance to levamisole and parental if we identified UNC63 into a worm, truncated UNC63 into a worm. And this is what we confirmed in vivo in the lab, selected HCO-RH6 that comes from Marlene Ross labs, that has been selected artificially for its levamisole resistance starting from a susceptible strain. So this strain has been selected only for levamisole resistance, had never been in contact with parental, and we demonstrated that in vivo the receptor, the worms are cross-resistant between levamisole and parental. So clearly now this has to be further confirmed in field isolates from Emanchus, but also as we also identify this truncated UNC63 in highly resistant isolates from Teladorsagia or Trichostrongylosis, this has to be checked also for this species. We have also to confirm that this truncated UNC63 is strictly associated with high level of resistance. And this is also of importance as this cannot be considered as a universal marker. Now concerning RCR8b, even though it has been associated with levamisole resistance in many field isolates by different labs, there is currently no functional validation for these markers. But at least what we can do is to investigate potential RCR8b in other levamisole resistance from other species to see if we are keep going on confirming that this might be a marker of interest. But because levamisole, parental and merontel could have differential or preferential targets into the worms, depending on the history of the treatments, this also means that there is potentially still a chance for parental or merontel to remain active on levamisole-resistant parasites. And clearly what it suggests is that instead of monitoring anthelmintic resistance as the only goal, we should really start thinking about also monitoring anthelmintic susceptibility. Because levamisole, parental, and merontel side resistance have been described in field or laboratory isolates from many species. Clearly, it was commonly accepted that resistance to this cholinergic agonist was co-inherited. But in sharp contrast, our results suggest that levamisole, parental, and merontel cross-resistance might not be inevitable. So, supporting this hypothesis, other studies have already reported that in field or lab-selected isolates of at least Trichostrongylus, merontel-resistant worms remain sensitive to levamisole. And in our lab, some of our own strains, we showed that some multi-resistant worms that are resistant to benzimidazole, ivermectin, levamisole, and parental remain susceptible to merontel. And they are also accumulating evidence for potential levamisole reversion. In some of our strains, if we do not maintain levamisole pressure, selective pressure on these worms, they come back to levamisole sensitivity. So clearly now what we need is a tool to monitor at the same time both susceptibility and resistance to levamisole, parental, merontel, but also surely all the other anthelmintics in a wide range of field isolates. And in that respect, what we would like to do is to take advantage of the automated larval migration assays that have been recently developed into our lab by Fabrice Guignard. So this technology was first developed for quantifying RNAi phenotype on Haemonchus contortus. So this technology is based on a spectrophotometric approach that is used for the monitoring of the larval stage motility. And this is real time. Here, just some example of the curves you can get from a susceptible isolates and a multi-resistant isolates. And here in black, I mean, this is the accumulation of fluorescence that is correlated with the migration of the larvae into the recording chamber. And you can see that you can detect slight difference of motility depending on the dose that you are using. And just to give you an example here, with parental, with 0.75 micromolar, you are almost completely paralyzed. Whereas to get the same phenotypes, you need to use 64 micromolars for parental. And the good news is that this is also working for ivermectins. And here for 0.02 for the complete paralysis, here for the complete paralysis, you need 0.32 micromolar. So this technology is very attractive because it can allow you to estimate quite quickly the EC50 and also the resistance ratio, here, for example, for parental. But it's also very attractive because it can allow you to identify some active compound to control the resistant worms. And for example, here are some drugs that are as efficient on both the susceptible and multi-resistant strain. So what we are doing right now is that we have already successfully adapted this technology to a wide range of anthelmintic drugs, including MLs, so that is very important. But also we have adapted it to a wide range of parasitic species from the different clades, and it also works on plant parasitic nematodes. And what are the work that are ongoing? Now we are trying to use this assay to detect resistance into mixed species that is more relevant with what we found in the field. So clearly, it is going to be very useful to evaluate the robustness of the different molecular markers we already have into your hand and the one that will come in the next future. But this will be also a very attractive decision-making tool for the field in the near future, something that will be very complementary to the fecal egg count reduction test. So to finish, I would like to acknowledge most of the present and past members from our team, as well as all the collaborators that are involved in this project. And I would like to thank you for your attention. And if I can answer your question, it would be my pleasure. Many thanks.
Thank you very much, Cedric. Excellent talk. And I can see from the Mentimeter some questions. So we start with the most voted one. How do you suggest to monitor efficacy and resistance in the same time?
So the answer is quite simple. At the starting point, I mean, we were using, single runs of larvae. So you had one batch of larvae, one drug, and you were testing, okay, how does it migrate? And then you get your answer. It takes something like 15 minutes. Now the test has been adapted to work with high throughputs. So we are able to perform based on the collaboration with INRAE. The technology has been adapted to 96 and even 380 well plates. So at the same time, you have your batch of larvae. You will spread halicots of your larvae on all the plates, and you would just need to distribute different concentration of the different anthelmintics. And you will have your controls, susceptible worms, resistant worms, and you will exactly see how they behave, and you will be able to determine with the same sample which drug is remaining active on the worms and which drug is not active anymore. And this will be very informative because we will be able to detect potential cross-resistance, and you will be able also to help the vet for the decision by saying, okay, you've got this resistance, but your worms are still susceptible to this drug. So maybe it might not be applicable because you might have some constraint because of your lactating animal or something like that, but at least you can have a good overview. And because it has been adapted also to high throughputs, you can also imagine that you can make this diagnosis at the ship level, because you only need something at about 300 to 400 larvae per run in the plates. So if you got enough fecal material, you will have enough larvae from a single sheep to describe all the sensitivity and all the resistance at the single animal level. That will be also very useful. Or in contrast, you can think about pooling your fecal sample and have a rough idea about what is occurring in one place.